Next up, PI presentation time. So we've got three very special guests. I think you guys are going to enjoy the presentations today. And first up, we have Dr. Alessandro Grattoni from the Methodist Hospital Research Institute. Dr. Grattoni is a repeat customer of National Labs, so we're happy to have him here today. He's going to tell you about what he's done up to date, and it's interesting, Dr. Grattoni actually has his payload, his experiment on station as we speak right now. So with that, Dr. Grattoni.
Well, thank you for this invitation. Actually, I take the opportunity for thank Casey's and all the collaborators on all the projects that we have flown on the International Space Station. So this has been extremely a great experience. We received several funding from Casey's, and thanks to this funding and thanks to matching funding from the Houston Methodist Research Institute, we were able to put together a Center for Space Nanomedicine. I'm chairing the Department of Nanomedicine, which is one of the largest departments that focuses on nanotechnology, development of nanotechnology for medical application. The department was founded by Dr. Mauro Ferrari, who was one of the pioneers in nanotechnology for medicine. What we do in our department is we take a number of different disciplines, we merge them together. So we're talking about engineering, physics, computational science, material science. And then we go talk to the doctors. We are connected to the hospital, so we actually just walk across the corridor. We go and talk to the clinician, ask them what their challenges are. And we apply all those disciplines and our nanotechnological approaches to treat diseases of various sources. We're talking about cancer, diabetes, infectious diseases, and so forth. But thanks to the Center of Space Nanomedicine, now we utilize the infrastructure of the International Space Station to do this type of development as well. Today, my story is going to be the story of an implant, a tiny implant. So this implantable device is a drug delivery system that can be inserted subcutaneously, for example, in the inner arm, as the schematic shows, and can deliver drugs in a controlled fashion, either in a sustained manner or in a tunable manner, remotely controlled. But this implant has big implications, not just in medicine, but also in the oil and gas industry, as well as in potential space exploration. Let me start from the medical application. So we're using this implant for HIV pre-exposure prophylaxis. So for the delivery of therapeutics that may prevent people from getting infected by HIV. We are collaborating with major pharmaceutical companies such as Gilead Sciences, who has provided us with funding together with the NIH to study and develop this implant in a way that can be transcutaneously refilled so that once implanted can be utilized for months into the patient, and when the drug is completely gone, can be transcutaneously refilled in place of being extracted and inserted with a new one. So we were able to demonstrate in macaques and on human primate studies that the implant is capable of achieving preventive level of the metabolites of the two drugs. And so this is a very effective manner to deliver HIV pre-exposure prophylaxis. Another application is the use of this device for the constant and sustained delivery of drugs for metabolic syndrome and obesity, for the treatment of these two kind of diseases. And we were able to demonstrate once again in non-human primates, in obese non-human primates, that this device was able to deliver GC1, so Betirom is one of these pharmaceutical compounds, that it was able to reduce the body weight of the animal dramatically, about 35% in this spontaneously obese macaque, without side effects, without cardiotoxicity, which are commonly associated with this type of weight loss medications. But what is special about this implant? What is the key of this implant? Well, we utilize nanochannels. So we utilize the nanotechnology and nanofluidic system. We use channels that are as small as 2.5 nanometers, so very, very tiny. Just think about the size of a protein molecule. And we manufacture them with the same technology that you use to make computer chip. And we manufacture them at the industrial level already in collaboration with a company called Nanomedical System, which is located in Austin, Texas. So this 2.5 nanometers, just to give you an idea, is about 20,000 times smaller than our hair. And the reason why we need this is because we realized that if we use these tiny, tiny spaces, membrane with these tiny channels, we are capable of achieving constant delivery of drugs without any sort of pumping mechanism, without any sort of valve. I'm sorry. So the drug is released out of a reservoir, an implantable reservoir, in a steady manner until the drug is completely gone from the reservoir. But additionally, if we apply an electric field across this nanochannel system, we can modulate, activate, deactivate, increase, and interrupt the drug release from the implant. Again, with no pumping mechanism, it's just through remote control via Bluetooth. So what happened at the nanoscale is there's a number of new phenomena that can be actually investigated. And what we did in part of our research was investigating exactly the diffusive transport in these tiny confinement spaces, as well as the gas flow, so which are very important for a number of different applications, as in the case of natural gas extraction, so for the oil and gas industry. Matter of fact, we have received a lot of interest from oil and gas companies such as Shell and Exxon. However, to study these systems and these new physical phenomena, we had some challenges. In fact, you're dealing with a space that is very tiny. Really, we are talking about the size of molecules, and therefore, you cannot utilize a conventional microscopy technique that you would do, for example, studying cells and biological systems. So we had the opportunity to propose this project to cases, and we were funded to study a surrogate model of our nanochannel system, creating a macroscopic scale of our device, using microchannel and microparticle in place of our molecule into nanochannels. And we were able to replicate what we observe again at the nanoscale in 1,000 times larger scale. So we studied this. We received also some interest from the media. The Houston Chronicle put us on the first page. We were using the infrastructure already available on the International Space Station, the microscopy model. We were capturing images on chips possessing nanochannels of this fluorescent microparticle diffusing from one side to the other, from one reservoir to another. And we were collecting images and bringing down back here where we did particle tracking and started doing all type of theoretical analysis of the results. Although this work is still ongoing, we know that we have some level of success, and we hope to be able to demonstrate it with the publication that is going to come out soon. One of the two publications is coming out of this work is being considered in one of the main Nature journals. But importantly, also, we are seeking additional opportunity to refly this very same experiment because of the intriguing results that we have achieved, and also because we want to expand the set of data collected. We took the implant, the very same implant that we use on her for medical application, and we inserted in mice, and in collaboration with Novartis, we tested these little devices for the delivery of a drug for preventing muscle atrophy. This happened on December 15. So the idea is that, of course, astronauts, they are exposed to microgravity. If they don't exercise, they tend to lose muscle, so muscle wasting. And this happens, of course, in animals in a very accelerated fashion and very acute fashion. So that makes mice model of muscle atrophy on the International Space Station ideal model for testing mitigation technologies. Importantly, down here on Earth, muscle atrophy is suffered by many people in the form of cachexia, people that are at bed rest for extended period of time due to, for example, debilitating surgeries or debilitating chemotherapy. So it's a real problem that affects a number of people, especially in the elderly population. At this stage, we have received a first, after flying the animal on the International Space Station, the first cohort of animal have come down already, and we have already started analyzing them. And we are waiting for the second part of the animal that were on the International Space Station to complete the study. But so far, everything has been successful. Thanks to cases, a third project was funded for the development of the next generation of the implantable system, an implant that can be remotely controlled via Bluetooth. Of course, the repercussions are multiple. This implant can be remotely controlled by the doctor from the doctor office while the patient is at home. So it doesn't have to travel all the way to the doctor to be potentially checked. It can be done in a telemedicine type of approach. And this, of course, has implication or application down here on Earth, but as well as for space exploration, where, of course, may be complicated to treat astronauts otherwise. We have developed prototype of these implants. These are actually existing and functioning. This was one of those. This that you see on the left is the newest generation of devices that we have tested in non-human primates model. And we are ready for flight on the International Space Station by means of demonstration into mice. So we have implants capabilities with devices that are small enough to fit into mice. Now I want to switch gears a little bit and talk about materials. Of course, materials are at the foundation of any new technologies. And interesting, Automobili Lamborghini, of all companies, came to us, the Houston Methodist Research Institute, and asked us to perform a biocompatibility study of 16 proprietary carbon fiber material they have developed. This carbon fiber material can be 3D printed, can be forged. So with manufacturing techniques, they are not very conventional to carbon fiber materials in general. So they gave us about $1 million to do this testing, testing acute toxicity, subchronic toxicity, and chronic toxicity over a period of 3 years. And I will come back to the partnership with Automobili Lamborghini, but this is to introduce the work that we're doing in material science. Matter of fact, on August 14, in collaboration with John Nicholson, Jason Sakamoto, and Dr. Cortiella, we flew an experiment on the International Space Station where we were looking at the recellularization from mesenchymal stem cell of tissue that was decellularized. This was lung tissue that was decellularized prior to launch. The interest was in understanding better wound healing purposes, but also in developing new technologies such as this one for cell transplantation. We're using combination of 3D printing and, again, decellularized tissue to create an environment that can be subcutaneously implanted, that can host transplanted cells, as is the case for pancreatic islets. Pancreatic islets that deliver insulin in response to a glucose stimuli. So the main idea is to create an environment that can be inserted sub-Q, it can accept various type of cells for a number of different applications, such as, again, diabetes or hypogonadism and others. We were able to, down here on Earth, to demonstrate, of course, the efficacies of this system, but we are awaiting to complete the study that was performed on the ISS to develop better ways to encapsulate this system with stem cell regrown tissue prior to insertion in the body. Going back to the carbon fiber materials, what we are intending to do with Automobili Lamborghini is utilizing, again, 3D printing capabilities to develop new tools for internal fixators for orthopedic application, as well as maxillofacial reconstructions, which are really, really big in of course, in orthopedics. But most interestingly, they are also very kind of puzzled by the opportunity of using their material into aerospace application. And they are providing us with 5 different very promising carbon fiber materials. They want to be tested on International Space Station. And this is kind of the topic of one of the next funding proposal that we will send to cases for consideration. And with this, I conclude my talk. And this, I want to acknowledge, of course, the work of cases and the great partners, such as Bioserve in particular, that has been our implementation partner since the very beginning. But also, I want to thank NASA for their support with the crew time and the help at the various facilities at Kennedy Space Center, as well as NASA Ames and Glenn Research Center. Thank you.
So Dr. Grattoni sleeps about an hour a day, and he didn't get to sleep at all today because he came here and spent his hour of sleep to talk to us. But if you notice, that's a great example of Dr. Grattoni and his team view the national lab as another tool in their toolbox, and microgravity is another tool in their toolbox. It's not one discrete experiment; it's a campaign. It's part of their institution now and how they do R&D, and this is the potential of the national lab and the ISS. So. And the partnerships, you saw the names up there that are involved in his various projects. So great job, Dr. Grattoni.